Transposons are devices for introducing new genetic material into chromosomes of all organisms. Transposons are segments of DNA that are capable of shifting from one location to another. A transposon enters the cell by being carried on a plasmid. It can move from the plasmid into the host cell genome.
A Sleeping Beauty transposon system is a synthetic DNA transposon designed to introduce precisely defined DNA sequences into chromosomes of vertebrate animals.
This transposon has been dormant for more than 10 million years in salmon and fruit genome, possibly suppressed by epigenetics. In the 90s, it was awakened by two scientists from Perry Hackett Lab in Minnesota. This is the reason for its name, Sleeping Beauty.
I know you, I walked with you once upon a dream.
Hello, it's me.
Vertebrate active transposons have never been isolated because although they are present, they contain a lot of mutations that inactivate them. But makes sense because the presence of active transposons could have awful consequences for the stability of genomes. The inhibition of transpositions may be due to external cellular factors or intrinsic factors as mutations in the transposase or in the inverted repeats. To obtain an active transposon, mutations need to be repaired. However, if the mutations of the transposon are repaired and it is introduced in a genome, we will find a massive transposition that will result in a genetic instability and therefore an interference with our experimental objectives. To solve this, we use a dissociated and heterologous transposon system. This means that the gene of the transposase has been separated from the terminal inverted repeats and that the host intervention is minimum. To solve the problem of the inactivation of vertebrate transposons, they have been repaired comparing sequences phylogenetically similar transposons and using directive mutagenesis to build an active transposon from inactive mobile elements isolated from salmon. This repaired transposon has been called a Sleeping Beauty. It is functional in mouse, fish, and human, and it results in a useful tool capable of stable integration.
Unbelievable! Unbelievable!
One component contains a gene of interest cloned between the transposon inverted terminal repeats, encoded by a plasmid. The other component is either a transposase expression plasmid or synthetic mRNA encoding the transposase. How it works. A Sleeping Beauty transposase binds to two direct repeats in each of the inverted terminal repeats of the transposon. The transposon is cut out of the plasmid and inserted into a target DNA, shown as chromosomal DNA. SB transposons, like all TC1 mariner type transposons, integrate into TA dinucleotide base pairs, which are duplicated on each end of the insertion site. In 2005, Adam Dupuy modified the transposon to turn it into a mutagenic agent. In the free space that the transposase left, a gene structure designed to produce mutation was introduced. It included signals of ending in both senses to stop the transcription of the gene where it is introduced, and also a promoter to induce gene's transcription from the integrating point. By this way, if the transposon jumps in an active gene, it will inactivate it. And if it jumps in the right orientation in an inactive gene, the latter will be activated. Therefore, we get the transposon to cause mutations in oncogenes as well as in tumor suppressor genes.
Over the past decade, Sleeping Beauty transposons have been developed as non-viral vectors for introduction of genes into genomes of vertebrate animals and for gene therapy. This introduction may occur in intergenic regions, producing the expression of a gene. This process is called gene transfer. Another possibility is that the introduction of the transposon occurs inside another gene, so its function is disrupted. This process is called insertional mutagenesis. For either gene delivery or gene disruption, Sleeping Beauty transposons as non-viral vectors combine the advantages of viruses and naked DNA, avoiding the defense machinery that host cells employ against vectors. Transposition into germline cells always takes place, being useful to make transgenic organisms and to identify the role of a gene or to identify cancer genes, knocking out their functions in both cases. On the other hand, in somatic cells, transposition does not always occur. A particular field is its use in gene therapy, a technique that treats a disorder by inserting a gene into a patient's cells instead of using drugs or surgery. Let's see a practical example. It is known that the USP9X gene is active in normal cells. In pancreatic cancer cells, this gene is inactive. But how can we confirm this evidence? The answer is easy, using the SB transposon system. If we make transposition of a genetic cargo inside an active USP9X gene, we disrupt and inactivate the gene. As a result, the cells that have suffered transposition undergo pancreatic cancer.
The Sleeping Beauty transposon system is an effective tool that has many advantages as a genetic tool, which include the following ones. Facility to obtain mutants, mainly mice. Embryonic stem cell manipulation is required. Non-invasive screening and rapid identification of disruptive genes are possible by using the transposon sequences as a DNA tag. Region-specific saturation mutagenesis is possible. Since the development of the Sleeping Beauty transposon system, several other transposon systems have been reported to be active in mammalian cells, including Minos, piggyback, and Tolcho.
The first reports on using the Sleeping Beauty transposon for gene therapeutic purpose are now published, and the areas and questions that have to be addressed are now being defined. The focal point of investigation in the near future are the improvement and optimization of Sleeping Beauty for efficient and safe gene transfer and a refinement of our understanding of molecular interaction between the transposon and the cell. The Sleeping Beauty transposes in a wide variety of cells, including somatic, germinal, and embryonic stem cells with significantly different efficiencies. But can Sleeping Beauty transpose in any cell, or are there limitations?
Houston, we have a problem.
Also, many questions remain to be answered in basic transposon biology. The collective work of several laboratories has established DNA transposition as a new system for gene delivery.